Okay, thanks, Natalie, and thanks to the organisers for giving me this great topic to talk about how to plan for EV research. So they did tell me I could take liberties to talk about what I wanted, and I started thinking about, well, is it about what you wear in the lab, the playlist, you come up with when you're planning your experiments? But I'm going to probably just be a bit more boring and talk about how you design good EV research experiments. So, planning EV research, how do you design experiments for working on EVs? There's so much literature out there now. Where do you start? How do you, do that initial search for planning different parameters for the experiments, and what is it you really want to understand? So I thought I'd start with just some really basic fundamentals around, as scientists, how we go about designing experiments. And the first thing is really around framing the question and designing a hypothesis. It's not about saying, I'm just going to go and try this and see what happens. You need to have an idea of what it is you're trying to do and how you're going to try and prove or disprove a particular hypothesis. And I'm going to talk in this talk about a few things. One, touch on about appropriate model systems, different models, different systems, how to try and choose the most appropriate model to use for your experiments. And also around measuring experimental endpoints. And I think this morning there was a lot of discussion around things like heterogeneity and how can you measure things. And I think that's a really key part of designing good EV research experiments. And also, which control should I use? This is really fundamental, and you really need to think about this from the outset. You don't want to hear back from reviewers from a paper saying, why didn't you do this or that? Because that's too late. So thinking about the controls, the model systems, your experimental endpoints are really key fundamental things you need to design appropriate EV research experiments. And the final point on this slide is about reading the literature, but reading it critically. And this is a graph from a student in the lab, Liam Hourigan, who's plotted the number of EV papers since the early 1990s up to the current day. And you can see at the moment, over the last few years, there's been about 6,000 papers per year published on EVs. That's 500 a month. That's a lot of material coming out. It almost looks as though we might have plateaued. So, 500 papers a month is still a great contribution to knowledge. And Liam, through looking at all of these publications, is estimating now that for every 3 data papers that are published, there's actually 1 review article. And I think that's an issue for our field at the moment as well, is choosing the right literature to look at for when you're designing experiments or looking at what's been done before you. So take a really good critical look at the literature. There's so much literature, especially, look how fast it's grown since about 2015. So I think looking at the literature critically is really key for designing good EV experiments. So EV research has, as we all know, some quite unique aspects. And I think EV research is obviously evolving as the knowledge base grows. We've seen so many papers coming out, so that's contributing to our knowledge base around EVs. And we've got things like guidelines, such as the MySEV guidelines, which first came out about 10 years ago, as everyone will know. And that's grown into what was MySEV 2018, which had just over 300 authors in the community, to the recent MySEV 2023, which came out very recently, which has over 1,000 authors. So the community has really been involved in developing these guidelines for the field. And it's really important to remember that things like MySEV are guidelines. They're not rules, they're not absolutes, they're guidelines. They're there to help guide doing good, rigorous, reproducible experiments. And these guidelines evolve with time. So if you compare MySEV 2023 with MySEV 2014, you can see as we've got more knowledge, we've evolved those guidelines. And an important thing about these guidelines is that they don't tell you exactly what to do, but they provide parameters for which you report. So that's how you communicate your results, and also things to consider when you're designing experiments. So again, take a good critical look at things like these guidelines to help you consider best practice, how to design those experiments, and also the kinds of parameters you need to report so others can hopefully repeat your experiments. And I think there's 2 major challenges for the field there's technical limitations. I mean, it's really hard working on things that are so small. They're hard to count, size, because we can't see them with the naked eye, that presents obviously challenges as well. And also mechanistic insights, which I'm going to touch a little bit on in talking about dosing and mechanisms of action. So I think these are two quite major challenges for the field. Technical advances might help us overcome those sooner than we think, but we have to remember that these limitations can lead us to make some quite big assumptions. So again, go back to the literature, read critically, don't assume everything. And also, sometimes there are inferred observations as well. So again, take a good hard look at the data in the area that you're looking at to make sure it matches what's being reported. There's lots of also complexities, hurdles, and gaps in knowledge. Well, with complexities, dealing with EVs, we're dealing with very complex biological samples. Some are simpler than others, but we are trying to isolate things out of complex biological materials, be it cell culture supernatants, tissues, or bodily fluids. And also, and this was covered a lot this morning, around heterogeneity. We all know it exists, but how important is heterogeneity to your experiments or what you're trying to do? So how sort of pure do you need your sample for your application? For instance, for something like a biomarker, you're looking for a robust biomarker. You might not need as pure a population of EVs to detect that biomarker than for perhaps isolating EVs for a clinical purpose where you need particular EVs with a particular property. So again, these are two other things to think about when you're designing your experiments. Some hurdles, well, EV experiments often rely on indirect measurements. So some of the instrumentation we use is measuring EV, say, size by indirect measurements. So that's something we have to remember as well. So also try and understand the technical limitations of the equipment that you're using in your experiments. And also, don't be afraid to challenge assumptions in the field. Science is about creating new knowledge, using new techniques. So because something might have been established for a while, if you discover something new and you've performed well-designed experiments with appropriate controls that challenge an assumption, that's okay as well. And there's also lots of gaps in knowledge, and I'll show a few in a moment. And that's why we're here at things like the Education Day and the conference this week, to exchange our scientific knowledge and try and fill some of those gaps that we have. So just a few words about model systems. So many of us will use cell cultures. So cells growing in a monolayer are very useful for certain kinds of experiments or for isolating EVs with particular properties, but they're quite a long way away from how cells really grow in vivo. So, you could consider using three-dimensional cell culture systems, organoids, for instance, and now we can, isolate EVs from tissue samples. That's been a development over the last 10 years or so, and also whole organisms. So, a cell culture monolayer is quite a way away in complexity from a whole organism, obviously. So remember the limitations of the model system that you're using. And particularly using things like cell lines, does a single cell type represent the true physiology for the experiments you're doing, the kind of knowledge you're trying to, knowledge gap you're trying to fill? And in terms of in vivo models, we do a lot of work with rodents, for instance, some transgenic animals, wild type. We know there's many, many different strains of wild type mouse, for instance. They have quite distinct properties depending on the kinds of experiments you're doing. So you do need to think hard about the kinds of animal models that you're using. And also to consider things such as sex differences, the age of animals, and also the environment that's applied experimentally to those animals. And just in terms of sex differences, a number of funding agencies now are coming out with statements around using both sexes of animals in experiments to try and decide if there are sex differences or not. And there's some statistics over here, this is from a few years ago, that 80% of studies in mice only use male animals. And that the sex of animals has an effect in biomedical research, and sex influences the course and severity of diseases like cardiovascular disease, autoimmune disease, neurodegenerative diseases, and asthma. So This is why you need to consider too with your model system, should I be looking at both sexes to really obtain the full biological readout for the experiment you're doing. And also, say if looking at biological specimens, storage differences, the source where you get the material from, and also collection parameters can obviously have very big downstream effects. So you need to consider those when you're designing your collection protocols if you're doing biomarker studies, for instance, as one of the parameters for designing your experiments. Something we'll talk a lot about at the meeting today and also throughout this week is around what kind of dose of EVs do you need to use? So what is a physiologically relevant dose of EVs? That's a big question that we clearly don't have an answer for, and it will again depend on the system you're using and even the condition, whether it's a disease or a model or a normal control, for instance. So we need to perform things like dose responses and time courses to look at those two parameters of using EVs and functional assays. And also think about the mechanism of action. It's all right to sort of apply EVs to a system and say, well, when I do this, this is the endpoint, but what is the potential mechanism of action? Getting an idea of that is really important to understand what's going on in your system. And these two figures come from a really nice review from a few years ago, which shows the range of doses. So they surveyed 50 EV papers that were applying EVs and functional assays, and they found you could get doses from 0.1 milligram of EV protein per kilogram of body weight, right up to a gram of EV protein per kilogram of body weight. So there's no correct dose that you can use. You can see there's a massive, massive variability here. And even within 3 different kinds of disease, cardiovascular, neurological, and inflammatory, there's also big ranges in the doses that you can apply. So we need to think about treatment regimens where you use single dose or multiple doses, site of administration, and also the limitations of the model system that you're using. And obviously, controls are very essential for these experiments. In terms of functional readouts, another big question, is cell death the best functional readout? If you apply a high enough dose of anything, you will kill your cells. So we need to think about whether this is the best readout. And again, thinking about mechanism of action. Cell death might be the readout you're looking for, but trying to understand what is leading to that is really important. And we also need to be able to test the specificity of the EV effects. So we need to assay background, the buffers, or contaminants, remembering we're trying to isolate something from a complex biological mix. So we need to consider there might be other things in there that are having the effect that we're seeing in our system. Also need to consider the physiological relevance of our model system. And again, highlighting the differences between cultured cells. Killing cancer cells in a dish is very different to curing cancer in a whole animal model. And so by developing mechanistic insights, that's really key to understand the underlying biology and the mechanism of action. So finally, just on the horizon, I think obviously there's clearly several current challenges with EV research. We're all here to learn from each other about how we can overcome some of those. I think standardization and rigor considerations are really important. And again, there's several publications looking at standardization and rigor efforts in a number of different systems for EV research. And that to really move the field forward, we need the continuous development of methods and technology, development of guidelines, also multi-center studies. We want to see our own research repeated in other laboratories. That's one of the best validations that you're onto something, is to see someone else in a different lab repeat what you've just done. And that leads into things like reproducibility assessments and development of education resources. And I've just highlighted a couple of figures from the recent MySEB paper, which shows that, if we look at extracellular particles as an entity, you can break them down into lots and lots of different things. Any one of these could be what's having your biological effect. So again, we're working with a very complex system. And also that no matter what system you use for isolating EVs, each one of these ends up with a mixture of something. So we really need to think about what it is that we're actually adding in our model systems and using for our experiments. So with that, thank you for your attention.